नमस्कार दोस्तों, वेलकम टू bizbotany। आज हम पढ़ेंगे सेकेंडरी ग्रोथ के बारे में। सेकेंडरी ग्रोथ, फर्स्ट ऑफ ऑल, व्हाट इज सेकेंडरी ग्रोथ? Secondary growth means what? Consider we have a stem and root. This is a stem and this is a root part. We have a stem and root. The increase in the length of stem and length of root, we call it as a primary growth. We are calling it as a primary growth. But some plant shows increase in the girth of stem and root as well as increase in the girth of stem and root. That growth we call it as a secondary growth. Because primary growth will be occur due to the presence of a meristematic tissues at the apex of the stem as well as root. Root apical meristem and shoot apical meristem. Meristem cells will continuously divide, which keep on adding the cells in the apical portion of the stem as well as apical portion of the root, which increases the length of the root and shoot. That growth we call it as a primary growth. But in some plants like dicot plants, they show increase in the girth of the stem and root. That growth we are calling it as a secondary growth of the plants. That will be particularly present in a dicot plant only because of the presence of a cambium layer in the dicot plants. Monocot plants don't have the cambium layer, that's why monocot plants don't show the secondary growth. Then how secondary growth will occurring in a plants? Consider we have meristematic cells. The main feature of the meristematic cells means they undergo a continuous divisions. They divide and becomes a 2 cells, 4 cells, 8 cells. These cells continuously undergo divisions. After the divisions, these meristematic cells, these are the meristems. Meristematic cells undergo a differentiation and forms a permanent cells. There are two types of permanent cells are present: simple permanent cells and complex permanent cells. In case of simple permanent cells, we have a parenchyma, collenchyma and sclerenchyma. And by these parenchyma, collenchyma and sclerenchyma cells, complex permanent cells will be formed, like a xylem and phloem. Xylem and phloem are formed by the component of simple permanent tissues. This process we are calling it as a differentiation. And in some cases what happens, these permanent cells regain the permanent cells. Consider these are the permanent cells. PC means permanent cells. They regain the meristematic activity. Meristematic activity means dividing cells. They regain the meristematic activity. परमानेंट सेल से क्या बन रहे हैं यहाँ पर? टेम्पररी सेल्स बन रहे, मेरिस्टेमेटिक सेल्स बन रहे. उस प्रोसेस को हम बोलेंगे डी डिफरेंशिएशन. ओके. परमानेंट सेल्स क्या बनेंगे? मेरिस्टेमेटिक सेल्स बनेंगे. उस प्रोसेस को हम क्या बोल रहे? डी डिफरेंशिएशन बोल रहे. देन फर्स्ट ऑफ ऑल वी नीड टू अंडरस्टूड द formation of a cambial ring. How cambium ring will be formed in the dicot stem and root. Formation of cambial ring. This is the structure of a dicot stem. What usually it consists of? It consists of outermost layer, we are calling it as a epidermis. The layers of cells which are present in between vascular tissues and epidermis, we call that region as the cortex regions. These are the vascular bundles which are arranged in the form of rings. Ring arrangement of vascular bundles is a characteristics features of the dicot stem. In dicots only you can see the ring arrangement of vascular bundles. What usually these vascular bundles consist of? Here, this portion, this blue colored portion, we are calling it as a primary phloem, and this red colored portion we are calling it as a primary xylem portion. In between this xylem and phloem, this green portions we are calling it as a vascular cambium is present. Vascular cambium. Vascular cambium, which is present in between these vascular tissues. Let me draw here. This is a vascular bundles. This is a cambium. Cambial cells. These green cells nothing but a cambial cells. Above the cambium we have phloem tissues. These tissues we are calling it as a primary phloem. PP means primary phloem. Below the cambium we have a primary xylem tissues. These are the primary xylem tissues. The cambium which is present in between the vascular tissues, we call it as a intrafascicular cambium. Intrafascicular cambium. This cambium which is present in between two vascular tissues, primary phloem and primary xylems, in between these two tissues we have a cambium layer which is made up of parenchyma cells. This cambium layer is made up of parenchyma cells. That cambium we are calling it as a intrafascicular cambium. Intra means in between. It is present in between these two vascular tissues. The cambium, the parenchyma cells which are present in the medullary region. What is medullary region means? The cells which are present in between two vascular bundles. These are the two vascular bundles. The cells which are present in between two vascular bundles, that region we are calling it as a medullary region. They are also made up of parenchyma cells. The medullary cells, these are the medullary raised cells. This region which is present in between two vascular bundles, we call it as a medullary rays. The medullary regions which is present adjacent to a intrafascicular cambium. This is a intrafascicular cambium. The medullary rays which is adjacent to a intrafascicular cambium, we call it as a interfascicular cambium. Interfascicular cambium. This inter and intrafascicular cambium parenchymatous cells, they forms a ring. Ring-like structures will be forming by a inter and intrafascicular cambium. That ring we call it as a cambial ring. The ring which is forming by the inter and intrafascicular cambium, we call it as a cambial ring. Next is the activity of cambial ring. Activity of cambial ring. What it doing? Activity of cambial ring. This is the cambium. This one portion is a cambium, and this one is a inter and intrafascicular cambium, which forming a ring-like structures will be forming. What are the functions they are doing? These cambium layers, what they are performing? We know that they contains a parenchyma cells. Parenchyma cells nothing but they are permanent cells. These are the parenchyma cells which are present in both the cambium. They are the permanent cells. They undergo, these cells, parenchymatous cells, undergo a de-differentiation process. De-differentiation process Dedifferentiation. Differentiation means what? Forming a permanent cells from a temporary meristematic cells. We call it as a differentiation. Forming a temporary cells from a permanent cells, we call it as a dedifferentiation. Here, these cambial cells, after dedifferentiations, they become a meristematic cells. Meristematic cells have the capacity to divide. These cambial layer cells are the meristematic cells. They gain the meristematic activity by a dedifferentiation process. These meristematic cells undergo continuous division. These cambial cells undergo continuous divisions which keep on adding the cells. The cells which are adding towards the primary xylem. Let me draw here. This is a vascular bundle, and we have this cambial cells. Consider we have one layer of cambial cells. A layer of cambial cell will be present, and now it is a meristematic cells. By dedifferentiation process, these cambial parenchyma cells converted into a meristematic cells. They keep on adding the cells by division, by meristematic activity. The cells which are adding towards the pith region, the center region, pith regions, we call them as a secondary xylem. The cells which are adding towards the xylem, primary xylem, we call it as a secondary xylem. The cells which are adding towards the primary phloem, we call it as a secondary phloem. Here we have a primary xylem layer, This region we are calling it as a primary phloem layer. Due to the adding up more layer of cells towards the primary xylems, it forms a mass of cells. This mass of cells crushes the secondary phloem as well as primary phloems and adds the more xylem tissues. This leads to the increase in the girth of the plant stems. That growth we call it as a secondary growth. This is something about the secondary growth in stems by the cambium layers. Thank you.